Over the next 2 years, a network of partners will create a three-dimensional atlas of the whole human embryo. The team, funded by the WSSS Human Cell Atlas extension, will generate an in-depth single-cell and spatial atlas of human embryos. My involvement in this project is focused on the dissemination of the datasets through publicly available web portals, and these web applications will enhance the utility of the data by providing an interactive resource for global research, clinical and industry partners. Spatial genomics is a new frontier in molecular biology. The emerging techniques allow scientists to get transcriptomic data and know the positional context of those cells in a tissue at ever-increasing resolution. Designing a web application that presents spatial data in an intuitive and useful web interface involves significant software engineering challenges. I work in the research software engineering team within the Hanifa Lab at Newcastle University, and will be collaborating with the OME team in Dundee and leading experts at the Sanger Institute. We are at the very start of this ambitious project, but the cross-disciplinary technical team will deliver with a strong focus on making the scientific data fair. That's findable, accessible, interoperable and reproducible. The FAIR guiding principles for scientific data originally proposed in 2016 have been embraced into best practice within the research software community. In a highly innovative scientific field, adopting fairness enhances transparency and helps the community converge on standardized methods and data formats. As we embark on this project, we have focused on an existing software tool called Vitesse. The visual integration tool for exploration of spatial single-cell experiments is a React-based tool developed by the Gellenborg Lab and supports both spatial and non-spatial representation of cell-level and molecule-level data. It leverages DecGL, which is a WebGL-powered framework designed with a layered approach for data visualization of very large datasets. Vitesse supports OME-TIFF, which is a TIFF image containing an OME XML metadata block in the file header. This provides all the compatibility of TIFF whilst allowing us to take advantage of the OME data model. Vitesse then leverages the OME-TIFF tiler Docker image to convert the image into tiles for each image plane, allowing efficient viewing, panning and zooming of large, high-resolution images. The team is in the process of preparing an initial deployment using early project data from the lab, and we are excited to scale this up as more data comes down the pipeline.